Good afternoon, good morning, and welcome to this HDSA research webinar where we are going to be hearing today about the PROOF-HD study of pridopidine. I'm Dr. Leora Fox, Assistant Director of Research and Patient Engagement at HDSA. And also on the line is our Chief Mission Officer, Dr. Eric Johnson. And I'll be introducing our speakers in just a moment, but I wanted to first give some reminders about how you can ask questions and just let you know about some upcoming HDSA research content. So we're going to wait until after the presentation to answer questions, but at any time you can type your questions into the chat box in your Zoom toolbar. This is going to be a closed chat, so only the presenters and HDSA will be able to see what you have asked. We've got a big audience today, and we're going to do our very best to get to as many questions as possible. I also want to let you know about an upcoming research webinar, HDSA research webinar, in which Picnic Health and Sage Therapeutics will be talking about how you can contribute de-identified medical records to help advance HD research. Picnic Health is going to talk about their technology that can help support people with HD in their medical care. So please check that out on Wednesday, September 21st at 12 p.m. Eastern. I also encourage you to check out hdtrialfinder.org to learn more about currently recruiting clinical trials and hdsa.org/blog to stay up to date on what's going on with HD Research week to week. That's written by my colleague, Kelly Andrew. We are recording this webinar today, so if you want to revisit this presentation or send it to someone else, you can find it at hdsa.org/research webinars, or you can go to HDSA's YouTube channel. You can use the button at the top of our homepage, and usually it'll be up in a day or two. And now I'm very pleased to introduce our speakers, Drs. Michael Hayden, Sandra Kostyk, and Andrew Feigin. Dr. Michael Hayden is the CEO of Prilenia Therapeutics and a professor at the University of British Columbia. He is very well known for his work in HD and other genetic disorders, has published around 900 papers, seen many drugs through approval, including the Korea treatment Austedo, and has founded 5 biotech companies, including Prilenia. He's won many prestigious awards, including being named one of the 50 Canadians born in the 20th century who've changed the world. And he was recently recognized as a Pharma Voice 100 Legacy Leader. This is a global life science award celebrating a lifetime of achievement and his inspiring and transformational leadership on behalf of patients. Dr. Sandra Kostyk completed her MD and PhD at the University of Chicago and her neurology residency at MGH. She's been the medical director of the HDSA Center of Excellence at the Ohio State University Wexner Medical Center since 2000, has participated in numerous Huntington study group, industry, foundation, and investigator-initiated HD trials. She's really dedicated to increasing HD awareness and education and improving care options for individuals and families affected by HD. Dr. Andrew Feigin is a professor of neurology at NYU and has been involved in HD clinical research for more than 25 years. He's run many clinical trials of treatments for HD and Parkinson's and is currently serving as a principal investigator of PROOF-HD. He has also served in several leadership roles for the Huntington Study Group, including chair. And in addition to his research activities, he maintains a busy clinical practice caring for patients with HD and other movement disorders. So thank you so much to all our speakers for being here. And I'm going to turn it over to first to Dr. Hayden to get us started. I think you are muted, Dr. Hayden. We can see your slides.
Yeah. Sec. Shouldn't be muted. Okay.
We can hear you.
Yeah. Okay. Okay, great. Well, thank you, and good morning and good afternoon to colleagues and friends and many people part of HDSA community and the Huntington community. It's a special honor and privilege to do this together with Andy and Sandra today. And I look forward to sharing some of the details with you about the Proof HD phase 3 trial. Just as a personal perspective, I've, of course, been involved in Huntington disease since I was a medical student intern here in Cape Town, South Africa, where I had numerous, and my PhD thesis was on Huntington disease in South Africa. And I was able to witness and visit families in their homes and witness the struggle, but also be imprinted by the resilience and the courage that all these families demonstrated. These were families often disenfranchised by apartheid, and also yet having tremendous courage and resilience and welcoming me into their home. This was just to go back. I've been in the field a long time, and here I am in 1979 with Marjorie Guthrie here. There's Nancy Wexler, and I'm at the back with a lot more hair next to the late Peter Harper as part of this. And this was the whole Huntington community at that time, about 35 people from around the world, but a very small community. Marjorie Guthrie played a key role and helped me to get into the United States and come to Boston. And so I'm very indebted to the Guthrie family and Marjorie, who was an inspiring leader and the person who founded HDSA in 1967. So Marjorie was always a source of great inspiration and support through my early years in the United States. So just to share with you, of course, everybody here knows about Huntington disease and both the clinical signs of manifest HD with motor impairments, cognitive impairment, and chorea. And when I was at Teva, I was fortunate to be able to develop and lead a drug to approval for chorea. But for families, the big issue is really losing function. And this occurs in the earliest phases, even before manifest HD, where patients lose their ability to manage finances, hug their children, go for walks, and manage activities of daily living. This was the second drug ever to be approved in the US, and this was deutetrabenazine for treatment of chorea, but that had no effect on total function. And for families, this is very much the key issue that remains. Decreased functional capacity is the major burden. This is emphasized in all interactions with families, where people become dependent on others for care. And there is a particular scale that measures this, which measures the ability to manage finances, take care of activities of daily living, and also taking care of themselves. Pridopidine is an investigation drug in both HD and ALS. It's given orally, and I'll come back to that in a moment, twice a day. It's safe and tolerable. And the past studies suggest that this may show maintenance of functional capacity. We know the mechanism of action now. It binds specifically and potently to the sigma-1 receptor. And there's some open label data that suggests that the effect may be enduring up to 5 years. I'm particularly excited about this being a small molecule because it has oral availability. It's easy to administer. It immediately penetrates the brain and all regions of the brain and has access to intracellular targets. Also, if there was any side effect, you can just stop the drug. And with a short half-life, this allows the treatment effects to be discontinued. Of course, it's lower cost and also doesn't need refrigeration. So it's an easy drug to administer, and hopefully, if successful, would be available and accessible globally. We know that this is now clearly, the target is the sigma-1 receptor. This is using a ligand that binds sigma-1 in the brain. It's called fluspidine. You see all the sigma-1 receptors light up, and then you pre-treat with your drug to see how much that signal is diminished. And what you can see here, when you pre-treat with pridopidine, which occupies sigma-1, it now completely blocks the binder to those receptors, indicating that this is the key target in the brain itself. The S1R, or the sigma-1 receptor, is a good target for treatment of HD. Firstly, it's highly expressed in those regions of the brain that are affected in HD. And here in the basal ganglia in the cortex, orange shows higher expression, very highly expressed in those regions of the brain most early and selectively impaired in HD. It also plays a key role in regulating crucial pathways and improving them, those pathways that are impaired in HD. And so you can see in Huntington disease, there are a whole host of cellular pathways that are impaired in the cytoplasm, in the mitochondria, in the ER or endoplasmic reticulum, and in the nucleus itself. And what this does is it regulates those pathways, and I'll show you just in a moment. And furthermore, there's some data, preclinical data, that shows that the loss of sigma-1 increases aggregates with soluble Huntington that may indicate worsening of the disease. So loss of sigma-1 increases aggregates. So here you have the data. Here's Huntington disease, a number of aggregates in a high power field. And this is in the absence of sigma-1. And here you can see aggregated Huntington compared to in the presence of sigma-1. So there are more aggregates associated with this. And these are from independent findings. This is a Japanese group from published some years ago. Now, when we look at the sigma-1, it binds and it's located on the mitochondrial associated membrane of the endoplasmic reticulum, a very important organelle in the cell, and it abuts the mitochondria, right next to the mitochondria, and that's mitochondrial associated membrane. When it's active by pridopidine, it improves mitochondrial function as measured by all of these features. In addition, when it acts on the cytoplasm, it also has impact on autophagy and increases BDNF, which is an important neurotrope or providing positive effects on neurons in the brain. It rescues the dysregulation of calcium levels, decreases stress, and also has impact on nuclear cytoplasmic transport, which is also important for neuronal viability. When you look at that, the end result is enhanced neuroprotection. Another way to show that this is important is when you delete sigma-1 using either deletion experiments in cells or in vivo in mice or an antagonist, what happens is these effects are completely abolished, which means that it's acting directly and intimately through sigma-1. So here you can see work from Chris Ross's lab, where you can see these are HD neurons from an animal model. Here you can see cell death in wild-type animals. Here you can see with the mutation, cell death is significantly increased. If you add pridopidine, you can normalize neuronal viability, restore it to what you see in baseline compared to normal. If you then take those cells and antagonize or delete sigma-1, what you see is you lose the effect completely. Again, showing that this effect is completely mediated through sigma-1. In terms of BDNF, a very important neurotrope in the brain, you can see that pridopidine here essentially increases BDNF up to 1 micromolar here. When you give an antagonist, you are able to stop it. This is a very selective antagonist, abolishing BDNF secretion. And also when you look at the mitochondrial function, this is just looking at an index of mitochondrial function, which is mitochondrial membrane. What you can see, pridopidine improves this. And if you then look at cells where the sigma-1 is deleted, but with the mutation, you lose the effect completely. All of this showing that the effect of pridopidine is sigma-1 mediated. And finally, here in the striatum, this is looking at striatal cells, and these are spines that impact on all the connectivity. Here you can see with sigma-1, you get a very nice increase in connectivity compared to animals that just have no sigma-1. And again, cross to sigma-1 knockout, you lose the effect completely. All of this showing the impact in vivo of this drug in the presence or absence of the sigma-1. So this was preclinical data. There was other data that also gave us some reason to believe in terms of the effects. TFC, as I mentioned, is an important measure of progression. It's accepted as a single primary endpoint by both EMA and FDA, and it's been deployed for over 4 decades. And here you can see that pridopidine actually stabilized TFC at 52 weeks, which was an exploratory endpoint in PRIDE-HD. And this was the first drug ever to show some impact potentially on TFC at 52 weeks. Now, in terms of motor function, it's important to state that in PRIDE, pridopidine failed to reach significance for the motor endpoint. And these results were because of unexpected and a long-standing placebo response. So there was no difference from placebo in that particular study. But further evidence that gave us some reason to believe came from neurofilament data. And neurofilament is a measure of neurodegeneration, and stability or reductions are associated with effectiveness in therapy. And what you can see here in PRIDE, it was an exploratory phase 2. We post hoc were able to get some plasma from patients that were available, and we did post hoc analysis. These were analyzed in the appropriate way using the appropriate technology. And what you can see here in placebo, in the placebo group, as the NFL went up, essentially the TFC went down. This is at minus TFC. So this showed the appropriate change for placebo, and again, adjusted, remained significant when adjusted for many different variables. When we looked at the placebo group in PRIDE, you can see that neurofilament went up in the expected way compared to, for example, change in neurofilament in TRACK-HD or other natural history studies. What was remarkable that the treatment group with 45 milligram BID showed some stability in neurofilament, which gave further promise to this particular trial. So here you can see it with 95% confidence limits. Firstly, the neurofilament never went particularly high and was associated with stability in TFC compared to the TFC changes from baseline, which went, the TFC went down as the placebo levels of TFC, levels of neurofilament went up. Now, it's important to note that even though this was very promising data, when we look at the likelihood of success based on industry norms, you can see here about 60% of phase 3 studies end up going to regulatory submission. 40% do not. So a priori, there is a 60% chance likelihood of success, but also a 40% chance of failure for whatever reason. And it's important to know that even though promising, this is not a sure thing. This is a phase 3 trial essentially being conducted to test the hypothesis. So it's designed to replicate and assess the beneficial effects observed in PRIDE. And we have a significant partnership in collaboration with HSG and our North American investigators, the PIs, Dr. Andrew Feigin, who was the chair of the HSG. Sandra Kostyk is now the chair of the HSG, together with, she's the co-chair. And our European investigators are Ralph Riemann and Ann Rosser. And so I will hand over to my colleagues in an effort to tell you more about this story. But let me just say that it's been, firstly, we've recruited, you will hear, recruited ahead of schedule with COVID, amazingly. And this has been a wonderful collaboration with HSG and also with TFS in Europe. But the conduct of the study has been exemplary, and we're very grateful to HSG and all the investigators around the world who have participated in this particular study. Sandra?
Unmute. All right, I'm unmuted, but it doesn't look like the video has me on yet.
No, you are on, Sandra.
Oh, okay, good. Sorry about that. I'm just going to go over a little bit of more details of how the study's been going, what's been going on. I think we're in a really important phase now. Enrollment's completed, and we're pretty much halfway through the study. And what's really key is that we continue to the very end to see if we can prove an effect here. So as Dr. Hayden had mentioned, the PRIDE study showed promise that pridopidine may affect the TFC, and proof itself is designed to directly take advantage of what we learned from all the other trials we've done and the trials that have been done in the past with pridopidine, with a population that's been picked and the studies designed very carefully to maximize the outcome and the likelihood of success of this trial. So some of the things that just to point out of how decisions have been made and what's been going forward is this particular slide shows the similarity between the patients who were enrolled in PRIDE HD, the early study, compared to placebo, using this exact same dose of medication compared to the population who's currently enrolled in the PROOF HD. And there were 499 subjects who enrolled. We enrolled early. We enrolled a little bit more than expected, so we're in a good place. Age, weight, other features, CAG repeat sizes were about the same. As you can see from this slide, that there's no significant difference. So proof is also, again, we're trying to take advantage of data we learned from the previous study, and I'll go in a few slides later on some of the details of why this population and the stage of disease is so important in our trying to achieve success with this particular trial. So in the PRIDE HD trial, there were a certain number of patients in stage 1 and stage 2 disease. In PROOF HD, there's a little bit more of a greater percentage of patients in stage 1 that should be to our advantage. Stage 1 is people who have TFCs from 11 to 13. Stage 2 is a little bit more advanced disease, but the disease tends to progress a little faster, and having a division like this may give us a better odds of having a success at the end of the study compared to progression in placebo. So we expect that there will be a bigger difference. So the study itself is fully enrolled. And that's an achievement. As Dr. Hayden mentioned, we did this during COVID. Our study population is dedicated. We've had very low dropout rates. People are making their visits, going out of their way. Our coordinators are doing a good job. This study's designed to replicate data from our other studies. I've had people ask me occasionally, well, if you know that there have been 3 other studies using pridopidine that show promise, why do we need to do it again? And one of the reasons is we need to follow FDA regulations, which have a primary outcome and prove that the agent's both safe and efficacious. And the primary outcome measure of the other studies, as Dr. Hayden mentioned, were motor symptoms and some other symptoms. The TFC was not the primary outcome of the previous trials. And the FDA, in terms of regulations and efforts of promoting and getting drugs to patients with disease, has been focusing more in the last few years on what aspect of the disease is the biggest burden to a patient. What do they want? What symptoms need to be treated? And this is a summary of the FDA voice of the patients, a series of programs that have gone on for years. And on the side of the study, it summarizes a statement from September of 2015, where the FDA itself is saying the UHDRS TFC is the most clinically relevant scale for looking at functional capacity in patients and most impact on their lives. And that's the measure that we'll be looking at as the outcome for this particular trial. Next slide. So I'm going to take just a few minutes to explain what the total functional capacity is. It's a measure which seems, it's a global measure, and it may not see it as fine as measuring how many seconds it takes you to tap your fingers in a minute. But what it does is it has an effect on an individual's whole life, and it correlates incredibly highly with disease progression, and it's been well validated. So it measures the effect that Huntington's disease has on your ability to maintain employment, help manage your finances, just do routine jobs around the house and chores, making meals, and then your ability to take care of yourself, do daily activities, ability to shower, to manipulate objects, to feed yourself, and how much support you need. Scores range from 0 to 13. And this next slide is, I think, a key point to make for looking at this study is that we have a lot of data over many years using this particular outcome measure. And what's key here is that you can see that the scores over time, as we've been following patients for years, and this particular paper actually came out about 20 years ago, but it's only been further validated over the clinical trials we've done over the years, where the score of the TFC goes down over a certain point each year. And the greatest change we see using this particular measure is actually in stage 1 and stage 2, and faster in stage 1 disease. Next slide. So, and as we've mentioned, we have the benefit of a lot of past knowledge, observational databases, clinical trials we've tried before, where we can look and use that to characterize, and this data was used to design this particular trial. On the top of the screen, you can see that what the placebo effect, patients showed decline in all trials, observational, CREST, 2CARE, and PRIDE, with the PRIDE in particular showing at least a 1-point drop over the 12 months of the study. And if you go down and you compare all the other studies we looked at again on study drug, what you see at the very bottom is there was next to no change in the course of 12 months in the total scores of PRIDE patients on pridopidine twice a day. So what that gives us a hint and a hope that based on the data from the PRIDE trial, the placebo patients deteriorated by about 1 point a year, and the patients on pridopidine remained relatively stable. That's a big difference, a big effect. And the other part that gives us hope, and we don't want to be over optimistic, we're still doing a trial, as Michael pointed out. The majority of trials, many of the trials that we've tried have not had success, but not only do we want to affect things for 1 year, the goal here is to be able to help individuals with HD long term. We only have a little bit of data. The numbers here are small, but in the comparing trials like 2CARE to the Open Heart, which is an open label long-term follow-up study of pridopidine, you can see that individuals at the latest endpoint, highlighted in purple, had a 5-point decline over the period of time, 60 months, and less than half of that decline in the individuals in the Open Heart study. So we're optimistic, but we have to be patient and keep everybody enrolled and keep things going. I'm going to turn the podium over to Andy to discuss what's really critical in proving to the FDA to get this drug approved: safety, in addition to the efficacy we've been talking about.
Thank you, Sandra, and thank you, Michael, for your excellent introductions. As Sandra mentioned, I'm going to be presenting some little bit more detail about what we know about the safety and tolerability of the study drug pridopidine. Next slide. So as you've heard, pridopidine has been studied pretty extensively in prior trials in HD. And so we have pretty extensive safety and tolerability data already, even before doing the PROOF-HD trial. We're gaining more from the PROOF-HD trial as well. More than 1,300 subjects have received pridopidine, and on average for about a year or longer. So this is what we would call 1,300 subject years. There's a lot of exposure to this drug in prior trials, actually. So we know quite a bit about the safety profile of the drug. And most of that information is at the dose that we're using in PROOF-HD, which is 45 milligrams twice a day. And the bottom line is that the data shows that this is a very safe and well-tolerated drug, including up to long-term follow-up data in up to 5 years in some patients with Huntington's disease. And in fact, I think we could say that the side effect profile has been comparable to placebo in prior trials. Next slide. This gives you an idea of what some of the common side effects that have been observed, or I should have called it actually, well, I guess we'll call it side effects. But when you come to clinical trials, we usually refer to this as adverse events. I'll talk briefly about that, I think, on the next slide. But the most common side effects you'll see here are falls, nausea, cold-like symptoms, diarrhea, headache, and dizziness. And you can just see by looking through this slide that the rate of these events in the pridopidine 45 milligrams twice a day is in general comparable to what we saw in placebo. So PROOF-HD, as was mentioned, is already fully enrolled, and it's an ongoing global trial. It's a phase 3 double-blind placebo-controlled 2-arm study. 2-arm means that there's one arm that is randomized to receive placebo and another arm that is randomized to receive pridopidine 45 milligrams twice a day. 499 early Huntington's patients have been entered into the trial. And the primary objective is to evaluate the effect of pridopidine on functional decline at 65 weeks as measured by the total functional capacity. Patients who complete the double-blind study will have the option to enroll in an open label extension study and to receive the study drug in what we call an open label fashion, meaning that there's no placebo. So people who were randomized into the placebo arm in the double-blind portion of the study will have the option to take the drug during the open label extension, and people who are randomized to study drug will have the option to continue in the open label extension. This slide just shows the journey of patients who are participating in the trial, and we made this card to give to patients who are in the trial to show kind of just an overview of what the visits are, what they entail. For example, at week 2, you can see a little phone there. It says a phone call. Week 4, patients come in and see the study team, and you can go and you can kind of go through and you see that the study goes out. You'll see this. I'll point out a couple of the key points. The 65-week point is where the primary endpoint of this trial will be. So we'll be looking at the change in TFC to week 65 in the placebo group versus the treatment group to see if the study drug worked. But as the way the trial designed, without getting into too much detail, people who are in the trial initially will actually continue to 78 weeks until the last patient reaches 65 weeks, in which case everybody will roll over into the open label extension study. Next slide. So far, the retention rate in the study has been excellent. This is really a key issue in trials like this. It's great that we were able to enroll 499 subjects and do it over the course of a year, but for the success of the trial, we need people to stay in the trial and to stay to at least that 65-week point, optimally to the 78-week point if eligible. So that we can get a sense for whether the drug is working. And so far, we've been very successful at that. People have really shown a commitment, patients and families, and investigators and coordinators, all the people involved in getting people to come back for their visits have been very successful in getting people to continue to come back for the scheduled visits. And we have a 93.6% retention rate. So far in PROOF, we found what we found in prior trials, which the pridopidine has been safe and well tolerated. And I alluded to this earlier. In clinical trials, we don't call things side effects because we don't know who's on drug and who's on placebo. So we call events that occur adverse events, and then we assess in the end, did more adverse events occur of different types in the placebo group or the treatment group. But during the conduct of the trial, while we're blinded, we just call these adverse events. And so adverse events happen to people over the course of a trial that goes on for over a year. And so we measure these adverse events. We don't know whether they're related to the study drug or not. And we assess whether we think they're likely or not related to the study drug. And in this case, actually, of the 499 patients entered in the trial, 385 patients have had at least one adverse event. The vast majority of these adverse events have been mild to moderate. If somebody falls and twists an ankle, that gets recorded as an adverse event. If somebody has a urinary tract infection, that gets recorded as an adverse event. So you can see that the majority of patients do have at least one adverse event. We've had 51 serious adverse events. There's a very clear definition of what that means, but they've all been rated as not related to the study drug. There were reasons before going into this trial to be a little bit concerned about potential for some ECG changes, electrocardiogram changes. That this has not been borne out in the Proof HD trial. We have not seen any ECG changes so far. So we have a safety monitoring committee, looks at the data in an unblinded fashion, and they've deemed that there's been no concerns and that they've recommended continuation of the trial without any modification. These are the 30 sites in North America where the trial is taking place. And the next slide, you'll see the 29 sites in Europe and the United Kingdom where the trial is taking place in 9 additional countries. We can go on from there. The regulatory agency in the United States, the FDA, has granted fast track designation to pridopidine for the treatment of Huntington's disease. And this is important because if the trial works, if the drug works and the data shows that it's working, this will make approval by the FDA hopefully happen in a speedier fashion and make it faster and easier to get the drug to patients. Just a quick word about the open label extension trial of the Proof HD trial. Here's the inclusion and exclusion criteria. The patients have to have completed the double-blind study to be eligible to participate in the open label extension trial without important protocol deviation impacting efficacy or safety. Patients have to be capable and willing to provide signed informed consent. The open label extension is considered a separate protocol. And you have to meet all criteria required to move forward with the open label extension. So far, 32 patients have actually completed the trial, and all 32 have opted to continue into the double-blind portion of the study. And we monitor adverse events in the open label trial just like we do in the double-blind trial. And so far, there's been one serious adverse event in the open label trial that has also been deemed not related to study drug. Just a timeline of where we stand with the main trial, the double-blind trial, the phase 3 trial. We're now still in the third quarter of 2022, soon to be in the fourth quarter. We're expecting the trial to wrap up in the first quarter of 2023. There'll be a period of time where we do data cleaning and review of the data to make sure everything's in proper order. And we're hoping to have top-line results in the second quarter of 2023. So I'll stop there, and I just want to say a big thank you to Dr. Hayden and his team at Prilenia, the sponsor for the trial. I want to thank all my colleagues at the Huntington Study Group for their incredible efforts in conducting this trial, and with our partner TFS, who's helping with the conduct of the trial in Europe. I want to thank all the site investigators and coordinators for their tremendous efforts in getting this trial up and running, enrolled, and continuing to have patients come back. And then most of all, I want to thank the patients and families who've made an incredible commitment to this trial, and we owe you a debt of gratitude. So thanks to everybody, and I think we'll open it up to questions.
Thank you so much to all of our speakers, Dr. Hayden, Dr. Kostyk, Dr. Feigin. And I see that folks are putting some questions into the chat. I know there's a lot of great information there, so I'm glad that we have some time for Q&A, and we'll just dive right into it. So the first question that I'll pose to you from the chat is probably for Dr. Hayden, given that he spoke about the science and the mechanism behind the drug, including speaking about Huntington aggregates and autophagy. And this question is whether the prevention of aggregates of mutant Huntington and enhancement of autophagy could mean that this drug is also lowering or clearing out the toxic Huntington protein from the brain.
So this is a great question and one that's being investigated right now. We know that there's decreased aggregates, and we know based on all the biomarkers for autophagy, including the formation of the autophagosome, as well as various biomarkers that indicate autophagy is increased, and they are predictably increased with this. The impact on Huntington levels has not yet been assessed, but is a topic of discussion and investigation right now. Theoretically, there could be some decrease in mutant Huntington based on these findings, but we are in the process of looking both in vitro and in vivo in an effort to see what the impact that this has on Huntington levels, and particularly mutant Huntington levels, but we don't know the answer to that question thus far.
Thank you. The next question, oh, sorry, I think I might have interrupted there.
No worries, Laura. I was actually going to jump in and ask a question that came up in the chat too. Someone was asking, are there any potential drug interaction issues that you've seen?
I'll just provide. So firstly, there was a worry that the drug could prolong QT, which is a ECG finding that is associated potentially with heart rhythm abnormalities. We've gone back and reviewed all this, and also there's another trial in ALS, and in ALS, other drugs that have impact on the same receptor, such as Nudexta, there was some concern. So the FDA asked us to more closely monitor both for this in the ALS trial as well as the Huntington trial. I'm pleased to say there is no QT prolongation of any clinical relevance in either trial. So this is exciting and important. So antidepressants were included as part of this trial. Up to a certain dose, patients can be on various antidepressants. And again, we've seen no impact with the synergy. So the interaction with other drugs, certainly from a side effect profile, appears to not be a cause for concern.
Thanks, Dr. Hayden. We have a specific question from somebody who is participating in the trial, and maybe I can loop in some other questions sort of about timeline. So in particular, we understand that the more data is going to be available probably in the second quarter of next year. And this person's wondering whether the unblinding will happen. When will folks who are finishing up their participation in the trial know whether they received the drug or the placebo? So maybe you could talk a little bit more about that timeline, because I know that there are other folks kind of wondering about longer term timelines. Can somebody be in the open label extension for a very long time? Have there been people on this drug for example, 5 years or more?
Maybe I'll just start and then ask Andrew and Sandra just to comment. So when we get the results, which will be in the second quarter of 2023, at that point, everybody will know, and the sites will know who was on drug and who was on placebo. So that'll be at the time of getting the top line results. Of course, for the open label, everybody's on drug. How long the open label goes at the moment, we're going at least 6 months from the last patient, last visit, and going into open label. So this is at least till towards the end of 23. But this will depend on what we show in the data. And if this is successful, then we would like to extend the open label for a much greater length of period. But a lot of this will depend on the results. But over to Andy and Sandra.
Yeah, I'll just add, I agree with what Michael said, that I'll just add that we usually break the blind. That means inform patients about whether they had been randomized to study drug or placebo after the primary results of the trial have been released. usually in a matter of weeks. And the way we do it, a lot of people often ask, how do we go about telling patients and how do they hear? The way we've usually done it in Huntington study group trials is that we inform the sites about whether we provide the sites with a list of their patients, their subjects, and whether they were randomized to study drug or placebo, and then we rely on the sites to contact the patients and inform them. So that you should be in touch with with the site investigator or coordinator after the results have been released to find out whether you received drug or placebo.
So just to clarify a little bit, I think part of the question was when the person switches from placebo to open label, will they be told they were on placebo or study drug? And the answer to that is no. It's everybody will know that they're on placebo after the switchover, but they on study drug, but they won't know whether they were on placebo for the beginning until the end of the study.
Until all patients have ended, and not only ended, but until the results have been announced, actually. Yeah.
I've got a question here that I want to share with y'all. It's got to do with using TFC as an endpoint. And the question is, there are certain things, certain functional activities that can significantly impact that total functional capacity score, things like stopping driving or retiring from work. And there might be people who are living with Huntington's disease, say people in stage 2, where those changes can occur. And the question is, are you concerned that there might be an impact of the drug that is not apparent using TFC because people might be consciously deciding to retire or to stop driving? So things that people might have been thinking about for a while and have been preparing for. Could there be changes in TFC that are not that are being captured appropriately because those things occur, but are missing the effect of what the drug might be doing?
Well, I can maybe take a shot at that question. It's an excellent question. I think in trials like this, what you're describing can occur, and the advantage of the double-blind randomized design in a large number of subjects is that people who are giving up driving or people who are giving up work for one reason or another should, if it's truly randomized, and it should be equally represented in each group, the placebo group and in the treatment group. So that although there may be changes in TFC that are not due to an actual change in the disease at that moment, the representation of that should be equivalent in the two groups. And the study's powered to take that into account, basically.
Thanks for that, Andy. So we've got a couple of questions, again, related to timeline. And I don't know if this is something that you can speculate on in terms of the regulatory side of things, but several people are asking, what does the timeline look like in terms of if this is successful, when can people who aren't in the trial have access potentially to pridopidine?
Well, I'll just comment on that. So if you go from results, then you have to have a meeting with the FDA to really, for them to do their own analysis and to discuss. That's called a pre-NDA meeting, which is a new drug application meeting. This can happen usually within 3 months. Then if the FDA, and we are working now on the NDA essentially to have everything ready except the efficacy. So there's a lot of things that go into a new drug application in terms of toxicology, preclinical toxicology, clinical pharmacology, a lot of history that goes in. We're busy beginning to write that now and have this ready by the time results are present, so that efficacy results would come in, but with most of the NDA written. We would like to submit an NDA within 3 to 4 months, maybe maximally 6 months post results. And then if the data is compelling enough that FDA might give priority review, which means the drug would be approved 6 months later. So if you say April to say July, then 6 months later, early 24, and then this would be available essentially the next day. So we're looking at 2024. The exact timing would depend on the time of submission, the time for review, and all of this will depend on the nature of the results. So this will be early 24, exact time to be determined.
Thanks, Michael. I think there's been a couple of questions about the retention that you've seen on this study. Both, noted that that's a significant percentage of people who have stayed in through the study. And the questions are what you think, why you think retention has been so good. And then another question was why people might have discontinued.
Well, maybe I'll comment again just briefly. I think retention has been so good because of the amazing willingness and partnership with the HD community. HSG has also played a major role in supporting this and having strategies to maintain retention as it continues. So that's the other issue, is that the side effect profile is not severe. So it doesn't cause any serious adverse events that appear to be related to the drug. And that's also very important. These two factors play a key role together with the ease of administration. The oral administration of a drug with a low side effect profile, together with the recognition of the importance and the participation as partners of the patient community, have all played a role. But I'd ask Andrew and Sandra to maybe give their perspective.
Yeah, I agree. I mean, I think first and foremost, I would say the commitment of patients and families to trials for Huntington's disease, the understanding that we need to do these trials and we need to see them through to completion in order to advance the therapies available for Huntington's disease. So that's what I would mention first and foremost. I think, to be honest, I'm going to say, I think there's also a major commitment, obviously, on the part of the site staff, investigators and coordinators that interact with the patients and convey to the patients the importance of seeing this through, that we can't determine whether the drug is working unless people stay in the trial till the end of the trial. And then I also want to just point out that I think something unique about the way the Huntington Study Group does these types of trials, actually, which is that we try to make ourselves available through things like what we're doing right now, but in other ways to respond to patients and to treat each individual person who's in the trial as an individual if they're having second thoughts and trying to find ways to keep them in the trial. And so, I don't think we can rest on our laurels. We got to really push through and get through the next 6 months and keep people in the trial. It's really critically important. But I think those are been the key factors from my perspective.
And I think that the teams know a lot of these families. This is a generational disease, and the coordinators, the PIs, have close relationships with a lot of the patients. And I know even in some of the other studies I'm doing where, we discuss the placebo effect or how they're participating. A lot of times I hear patients, subjects say, I'm doing this no matter what, because my children are at risk. This is a family disease. So this is a disorder that has a lot of impact on the family in more ways than treating a disease like diabetes or other disorders. So our patients are committed to helping us make progress in HD, no matter what the outcome of the study is.
Thanks both. I'm going to ask a general question about access, because we've got sort of access questions from different angles here. So maybe you could speak a little bit to those issues and how Prilenia is considering things like right to try, considering things like the ability to bring this to a more global population or a population outside of the eligibility criteria, and also things like cost. I know that's a lot of questions wrapped up into one, but it would be great to hear your thoughts on that.
So let me start off. Of course, for an oral drug, that's rapidly absorbed with a low side effect profile, this is a drug that would be particularly useful for universal access. Now, how that happens and how this drug is commercialized is still a big topic for discussion, both in terms of cost and in terms of who commercializes the drug, who distributes the drug. I can say manufacturing is relatively straightforward, always a bit complex, but not complex like a biologic drug, for example. And I would hope, my hope and prayer, that if we do partner this drug or we do it ourselves, that the philosophical intent is to make sure the drug is widely available, and also in those countries around the world who have contributed so much to HD, from Venezuela to South America to Africa for me and other parts of the world. So the drug will be priced according to advice from payers, what would be acceptable. And this is research that's still being done. But we would like to see, and this is not proven, that the drug would be given at low cost to those regions of the world where, in fact, the resources to pay for it are not available. So this would be appropriately charged in the Europe and the North America, with some. Hopefully, we can persuade nobody to take any profit, but distribute the drug appropriately globally. This is all in discussion, both with ourselves and potential partners, and we don't really have, but I think that philosophical intent is very much, the principle is very much underlying how we're approaching this. Let me also say, because I've seen two other questions around pre-symptomatic, and we are planning a prodromal study. In other words, people destined to be ill but not yet ill. And this study, we have an advisory board coming up in the next few weeks. We're going to be choosing sites, defining that, and this would be a study that would commence after results in the early part of next year. We're also planning a juvenile HD study, which would also be something that would happen in 2023. But all of this will depend on the rigor and the essentially how results are convincing when we get those results in the second quarter of 2023.
Thank you, Michael. And I want to respect everyone's time here since we're at the hour now. So let's say thank you to all of our speakers once again, and we can definitely do our best to follow up with Prilenia and with our speakers with the remaining questions. But thank you everyone who attended today. Thank you so much to our speakers for providing a lot of clarity on Proof HD and the timeline going forward, and to all the participants as well. So thanks all, and hopefully you'll join us for our next webinar on September 21st. Have a great day, everyone.
Bye-bye.
Thank you all.
Bye.
Bye-bye.